Hi everyone, welcome to the first one of BES's impact presentations. These is supposed to be a series of presentations about our work and how some of that work in the field overlaps with some of our software development. And our presenter today is Dr. Jerry Kelly, who helps run our field operations and is our project manager across a number of projects. Over to you, Jez.
Thank you, Michael. Can everyone see my screen?
Yep.
I hope so. Can everyone hear me okay too?
Yep.
All right, wonderful. All right, well, good afternoon, everybody. So this is going to be the first impact presentation that we're going to do what I imagine will be a series of presentations, and hopefully we can do them routinely because I think they're valuable. So what we're going to be talking about today is vector surveillance and our role that we're going to be playing in the Pacific on vector surveillance. So without further ado, if I can change, I thought the first thing we need to talk about is what is a vector and what are vector-borne diseases. Vectors are essentially living organisms that transmit infectious diseases or pathogens between humans and or from humans to animals to humans. There's lots and lots and loads of vectors around. What we are talking about when we're talking about vector surveillance or vector-borne disease at the moment are mosquito-borne diseases in the Pacific. And primarily we're going to be looking at Aedes-borne, which is Aedes is a type of mosquito arboviruses, and Anopheles-borne parasitic infections. So Aedes mosquitoes transmit viruses like dengue and Zika, and Anopheles mosquitoes, the most common one and the most common one that we were going to deal with is malaria. Firstly, what are some impacts and challenges that we face, particularly in the Pacific with mosquito-borne diseases? As we all know, and I'm not going to go into too much detail about mortality and morbidity, but we know that mosquito-borne diseases are a leading cause of these in worldwide, mortality and morbidity. For example, malaria has long been known as a disease of the poor, and what they basically mean there is not so much the mortality factor, but the morbidity factor, in the sense that malaria is, if this is consistent in your population and your population are consistently sick, then you as a population or as a country or as a province or whatever fail to thrive. And it's been proven time and time again that this is a huge issue that we face worldwide. What makes it challenging to deal with mosquito vectors in particular are largely their bionomics and the fact that there's so many different types of mosquito species. And as a result, their habitats vary widely, their behaviors, and so the environmental behaviors and vary widely as well. And when we talk about those, we're talking about how they breed, how they rest, what their life cycles are, even their flight ranges, where they live, where they sleep, etc., etc. When we're talking about dealing with vector-borne diseases in the countries where they're generally endemic, like the Pacific, we also have additional population level challenges, and these include mobile and migrant populations, very remote and hard to reach populations. So populations that are living remotely, often moving and interspersing between each other. The countries that we often are working in, and again, very relevant to where we work, is there always limited resources and our health systems are constrained. And so this is where Tupaia fits in really nicely, and is a primary purpose of Tupaia is to help strengthen our health systems in these areas. And we also have huge challenges and really scary challenges that relate to mosquito-borne disease, and that includes emerging multi-drug resistant pathogens and vectors. So both the diseases and the mosquitoes can and do build up resistance to the types of insecticides that we use or the types of drugs that we're using to deal with these diseases. Now, what is surveillance? Because we understand what vectors are, what is actually vector surveillance? And so the first point here is this is a WHO definition of what vector surveillance is. And it refers to the regular and systematic collection, analysis, and interpretation of entomological data for the purposes of health risk assessment, as well as the implementation, monitoring, and evaluation of vector control. What does that actually mean? The best definition I've heard in my time working is surveillance is essentially information for action. And so that means we generate useful information to use that to act upon that information. And so that differs from reporting per se, and sometimes there's a confusion between surveillance and reporting. Surveillance is an active approach to capturing relevant data so that we can use that data to make change or implement an action based on that information. Again, that sounds obvious, but in reality, it's really poorly done. Not only in the Pacific, it is worldwide, including Australia. We do time and time again fail to actively pursue good surveillance and utilize that information. So everything that we do in Tupaia should be based upon this idea that we're generating and providing information that should be used to inform action. It's really, really important to sort of have that in the back of your mind anytime we do anything, really. I wasn't going to include this slide, but I thought it was relevant, particularly given the COVID world that we're living in and the concept of what disease elimination is and things along those lines, because we're starting to hear these words time and time again. But effectively, as countries move towards trying to eliminate a disease, and this is very relevant, particularly in malaria and a little bit relevant to dengue, is they basically move from different phases of different programmatic phases. And based on those different programmatic phases, their operational priorities are different. And in turn, their surveillance priorities are different. So for example, if you're in an intensified control stage, this is sub-Saharan Africa, a lot of countries are still trying to maintain their malaria burden, for example. And so they're getting thousands of cases reported per whatever reporting period. So they're not actually going to be trying to track and do contact tracing on individual cases because they don't have the capacity and it's not what they're trying to achieve. As countries just go down the line towards elimination, that's exactly what they're trying to achieve. They're trying to track individual cases and trying to prevent the local transmission of that case. So from the imported case into a locally sort of regenerating population or pattern of transmission. However, throughout all those stages of what we want to do, from intensified control to elimination or even eradication, what programs really, really need, and what they need to access accurate data and practical tools to generate useful information that actually supports program implementation. And I think I'm going to really emphasize this throughout what we're going to talk about today, because it's such an important factor that we really do poorly. We need to have useful tools and we need to have access to information to be able to make decisions effectively. And we need to do that so that we can ensure the appropriate interventions are delivered at optimal levels of coverage in our target areas. And to be able to do this, we need robust and good surveillance response systems to be able to swiftly locate and eliminate transmission reservoirs. Again, I highlighted these because all of these are relevant to what we do with Tupaia, and that is we're aiming to provide a really good surveillance response systems that are timely, so that when the information is reported, that we can see, or our implementers can see this information, that the information is accurate and practical, and that we can use that information, or our countries, our partners can use that information to both decide what is an appropriate intervention, and to ensure that when they do that intervention, they do it properly. And so an example of that is if we decide that household spraying is a good intervention in a particular area to halt local transmission of malaria, we want to make sure that when they do it well. Because again, if we don't do it well, if we don't do it effectively, at certain levels, it basically renders the intervention useless. So I just wanted to talk briefly about a spatial support system approach to vector surveillance. And so this is some of the work that I was doing in Solomon Islands and Vanuatu when I met Michael and Ez, and have been sort of involved with ever since, really. Basically, what is a spatial decision support system? It is essentially Tupaia. It's a it's a computer-based system that interacts and that incorporates a wide variety of information to help us make a informed decision when we have a geographic focus to it. And so mosquito-borne diseases and their interventions have everything about them essentially has a geographic focus. So, i.e., what is the spatial distribution of our vectors? Where are mosquitoes? Where is the population at risk? where is current transmission happening? Obviously, we can utilize key information, and I'm not sure if my little pointer will work here, but basically this is what we do in Tupaia. We can provide spatial data files, like our GIS data files, our country boundaries. Surveys can be routinely collected in here. Expert knowledge, what we're talking about expert knowledge here is we're not talking about fancy academics or anything along those lines. We're basically talking about our in-country implementers, our program officers, our surveillance officers, our field officers, and utilizing their expert local knowledge to provide input into our systems. We can output particular reports, and based on all this information in together, we can actually have an informed decision or a targeted action. So some examples of what we've done in the Pacific in the past about this is, and again, if we're relating this back to Tupaia, we conducted in our elimination provinces, so in certain provinces in Solomon Islands and Vanuatu, they are still pursuing elimination, and some they've actually achieved it. And as part of that, we did quite detailed baseline surveys, what is known in the vector surveillance world as geographical reconnaissance. And that is basically exactly what we do with our baseline surveys in Tupaia when we're talking about basic clinic data surveys and facility fundamentals. What a geographical reconnaissance survey would do for vector surveillance is it collects a baseline within your target area of all your priority features. And so that includes households predominantly. It would also include suspected mosquito breeding locations, potential locations, things along those lines. Rivers, like for example, rivers, access points, blah, 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 blah. So basically, we collect our baseline to find out what is where and how much of it is there so that we have our intelligence data to act accordingly at a later date. When we're talking about intervention management for vector control and elimination, some of our frontline interventions, as we all would know, are mosquito net distribution and household spraying. So where our systems and our surveillance systems can support this throughout all facets of the implementation process. That includes planning, implementation, and monitoring and evaluation. When we're talking about planning, so clearly we can map out our target areas, and we can utilize useful tools like risk maps and things along those lines to identify where we want to target our information. And I'll talk about the classification of transmission probably in the next slide. And we can also use that information to overlay in our baseline data to identify information that will help deliver that intervention. So for example, we know that we want to spray households within the 2 kilometre coastline of Tanner Island in Vanuatu, because we know based on our transmission foci that that's where the mosquitoes are and that's where our population is. So our information system can identify how many households are within those areas. We can break them into operation areas. Then we can use that to plan out how much insecticide we might need per operation area, how many mosquito nets we might need. Then we can use that to support the implementation. So we can develop household checklists or village level checklists that we can give to our spray officers or our mosquito net distributors, and so that they can literally or digitally check off the areas that they're covering or spraying. We can use that information to find out pockets where we've missed, and this is the most important part when we're talking about implementing interventions. So this goes back to optimal coverage of appropriate interventions. And so this example here, what you're looking down in the bottom right-hand corner, is in southern Vanuatu, and it's a real example of a spraying campaign in the early stages of the elimination project that we're a part of. Down here, you can see pockets of red. These are red households, which means that through the first round of a spraying campaign, they were not sprayed. This is really typical in the Pacific. You can see these little pockets for whatever reason pop up if you collect that information. Normally, they don't have that information. And so they will basically say an island is 100% sprayed, where we know that spatially there are these pockets of a lack of coverage. And what we found out by looking at our information was that the chief of this village actually refused entry from our spray officers into the village because he was fearful of the insecticide that was being used. And the science tells us that basically we can sustain a pocket of transmission here without, which can impact in these areas because we're not getting our optimal level of coverage. And so basically they utilize this information, our malaria officers utilize this information, and they spend about a year working with this chief and a couple of other chiefs that refused the spray campaigns to basically implement another intervention, which we would call behavior change communication, but effectively it is just providing some education and chatting and lobbying with the relevant people to really explain the benefits and the community-wide benefits of accepting this intervention. And this chief of this village, Ipakal, actually became a malaria ambassador after that program, and ended up going around the whole province, basically promoting the benefits of household spraying to support malaria elimination. And it's actually a really successful example of utilizing spatial tools in the Pacific to help eliminate malaria. And currently Tupaia province, which is the southernmost province in Vanuatu, is officially malaria free. And that means they have no locally transmitted cases of malaria. It still is susceptible to malaria because through imported cases, but they have by definition eliminated malaria, which is really cool. Another example, and this is a little bit more interesting, and this kind of relates to our WISH project in the sense that we also implemented the use of automated algorithms to automatically locate and classify what we called areas of interest and utilize those to conduct targeted response measures where we were finding transmission. Basically, this was a combination of the spatial and temporal distribution of positively recorded malaria cases in conjunction with key vectoral surveillance data, which included vectoral capacity. And so that if the mosquito, the vector is there, the potential densities, known breeding sites, mixed in with things along the lines of seasonal weather patterns and things along those lines. And we can use that to create an algorithm to define what we would call a transmission foci. And so if we look down on this map, basically these different colors are different pockets of transmission foci. If we look at the yellow pocket here, this is what was defined as a new potential transmission foci. And what this actually meant and what this told us was that we haven't had a locally transmitted case within this area, so within this community for, I can't remember, I think it was a period of about a year, but we recorded an imported case. And because we know in that area that the vector exists and the vector has a relatively high capacity to transmit the parasite, malaria, it was automatically defined as a new potential area. And then what new potential tells us is that the most effective intervention based on a set of standard procedures is to spray houses, basically. And so what our entomologists and what our vector control officers in the Pacific or in Solomons would say a fast knockdown, because we wanted a rapid response to basically stop the interaction between the mosquito and the human as fast as possible. Whereas down in the southern east corner here, a residually active area, so this is an algorithm defines this area as residually active, we have ongoing transmission and it's been ongoing for quite a while. So basically the most appropriate interventions there are not necessarily a spray or a fast knockdown mechanism. It is to target what our officers would call the factories. And so they would be going out to actively look for mosquito breeding sites or potential breeding sites and doing some larviciding and basically treating and trying to halt mosquito breeding in those areas. And so that's an example of how we can use vector surveillance data to inform appropriate interventions. I hope that makes sense. That's basically a brief overview of sort of how we can utilize vector surveillance and how we have in the past. When we're talking about scaling this is where Tupaia really has so much potential, because we were using ad hoc GIS-based tools that did not really have the capacity to, well, they don't have the capacity to scale because someone has to be there, someone has to manage these, and they're often a little bit complex. Whereas Tupaia, the whole idea is that we can scale really quite advanced, user-friendly surveillance response tools to provide relevant information, again, to help those who are implementing interventions make the best decisions possible. So Strive is a project that we're working on, and this is a project based in the Pacific, which I'm probably not the best person. Aaron's probably the best person to chat more about this if we want to later. But effectively, Strive's project is stronger surveillance and system support for rapid identification and containment of resurgent or resistant vector-borne pathogens in PNG. And so effectively, we're building a good surveillance response system to rapidly identify when disease occurs and use that to guide response to contain this disease. And we're collecting really cool information as well about resistance, so antimicrobial resistance to the pathogen, which is I think we're working on malaria, so the parasite, and I think there should be some work, or I think I was reading there's some work to be done with dengue, so the virus, the dengue virus, as well as insecticide resistance for the mosquitoes, which is something I'm more used to doing. And so this is an example of some of the dashboards that we have already developed in Tupaia with for Strive. So at the top left, we're looking at positive Plasmodium falciparum and malaria by health facility. And down in the bottom right, we're looking at febrile illnesses. So there's no number of people presenting with cases at a sub-village level, which is also really cool to get to that high level of resolution. Pacmozy is another project, and this is the one I'm mostly focusing at the moment, which is a Pacific mosquito surveillance strengthening for impact project. And this is a regional project that we're going to, it's just started, it's still in the planning phases effectively. It's been a little bit slow given some of the delays we've had with COVID basically, but it's a Pacific-wide regional project with a primary objective to support and strengthen country-led programs to implement best practice vector surveillance response, mainly for Aedes-borne arbovirus, so that's dengue and Zika, but as well as we're talking about malaria as well, where relevant. Again, this is actually, it's a really, really interesting and cool project because vector surveillance is something that has not been done well anywhere. And it's actually a really hard thing to do well, but it's a really, really important thing to do well if we really want to target these diseases and to make a positive impact. And so key components of this project are going to develop standardized practices and that ought to do vector surveillance, and I'll go through them in a second. And so we're basically going to be designing Meditrack surveys to support these standard and priority vector surveillance packages or approaches. Really, again, support enhanced data management and use for effective decision making. And so that'll be at a regional and country level. And so we're going to be developing relevant vector surveillance applications in Tupaia to do this. And then we're going to be helping build that capacity within the country level to actually utilize these tools and utilize these tools effectively. So below, there's a few countries where it's proposed to be done: PNG, Timor-Leste, some of the other Pacific Island countries like Solomons, Vanuatu, Fiji. You can read for yourselves. So key surveys that are going to be developed in Meditrack to support this is effectively we're going to be looking at how to capture our baseline data. So we're calling it a project metadata form, but it is effectively a geographical reconnaissance form. We're going to be identifying field stations to monitor these key indicators that we want to capture and map them and capture essential baseline data around those. We're going to be collecting adult mosquito. We're going to be designing adult mosquito collection survey forms to capture how many mosquitoes and their presence across or throughout the countries. Larval and pupae survey forms, so these are relate to breeding sites. Insecticide resistance, so again, we set up basically sentinel sites to monitor changes in trends to mosquitoes resisting particular types of insecticide. That's actually, it's a really, really important thing to monitor, and some other lab-based forms as well. So the type of dashboards that we'll develop, some relatively simple ones at a regional level, but then we're going to start to develop some really, hopefully, some really cool country and sub-country level dashboards and map overlays that really help to target our surveillance and response intervention. So simple ones, but that are actually really effective at a regional level is: does the mosquito exist at a country level? So does the Anopheles mosquito exist in Tonga? The answer is no. So that means there's no malaria in Tonga. But does the Anopheles mosquito exist in Solomon Islands? Yes. Does the Aedes mosquito exist in Solomon Islands? Yes. So they're susceptible to both malaria and dengue. And these things can change over time, but it's not super common. Then we can go down to lower levels at the sub-country level. So for an island like Reno Bologna in the Solomon Islands, does Anopheles mosquito exist in Reno Bologna? No, it doesn't. So they're actually not that at the moment. It doesn't, and there's no trace of that happening. So they're not susceptible to malaria in Reno Bologna province. Whereas in Guadalcanal or Malaita, absolutely it does, and so does the Aedes mosquito. And so they're susceptible to both dengue and malaria. Down the bottom, you can look at insecticide resistance. This is, again, these are just mock-ups at the moment, and the types of insecticide classes and whether they have any proven or shown insecticide resistance. And so the classic example here, and I think this is actually, I find this table really interesting if we're going to go through it, but insecticide resistance as an example it is unbelievable, and this happens in Africa, in Asia, and in the Pacific, that countries will procure an insecticide, and they have done so for years and years and years. And that's the reason why when research and field work are showing that mosquitoes are becoming more and more resistant to this type of insecticide, but because we're not utilizing these data to make effective and appropriate decision choices, countries will be procuring the wrong insecticide and utilizing this for both spraying and mosquito net distribution time and time again. And it's just one example of sort of how we could easily change our policies, change our way of thinking by effectively utilizing some really relatively easy information to capture, and certainly easy information to report on, to really make some positive and impactful programmatic change. But just briefly, here's some examples of core surveillance indicators. And basically, I want to show this table so we can get everyone thinking about how can we present these data. Firstly, how can we capture these data in our surveys, and how can we present these data via Tupaia in a way that our partners in the country can really utilize this information, because that's what's critical. Adult mosquito occurrences we just chatted about, it's fairly obvious. Insecticide resistance, the same thing. It's important to select insecticides to which the local vector species are susceptible to. So it's relevant for indoor spraying. LLIN and ITNs is mosquito nets and larviciding. So when we're basically trying to kill the pupae in the breeding sites. Resting habits is also an interesting one, because mosquitoes have different resting habits. Some prefer to rest indoors inside your house, some prefer to rest outside your house. And so if you're, so an Aedes mosquito example, so an Aedes aegypti mosquito, which is a dengue mosquito, often doesn't necessarily rest indoors. So is there a benefit to do indoor residual spraying to combat that mosquito? Knowing the resting habits, and these resting habits can change, along with the feeding habits. When and where do mosquitoes feed on, particularly on humans? And so I'll show you an example of this. So at the bottom left-hand corner here is a colleague from the Solomon Islands. This is Professor Hugo Bogoro. I think there's a Michael probably know him. So what he's doing here, this is what is known as the gold standard of adult mosquito trapping, and it's called human landing catching. And what he will do is sit there all night waiting with his legs exposed like that, waiting for mosquitoes literally to bite his leg or to land on his legs, and hopefully he sucks them up in that straw before they bite him, so they can capture when and how many mosquitoes bite throughout a day and night period. And so that way we can work out mosquito biting patterns. So does a mosquito bite early in the evening? Does it bite in the middle of the night? Does it bite at dusk? Does it bite during the day? And so that obviously tells you information like, will a mosquito net be effective? And the answer is, if it's in the early evening and no one's actually underneath the mosquito net, so maybe it's less effective than what a repellent might be, for example, or potentially insecticide treated clothing and things along those lines. Obviously, there's some serious issues with human landing catchers, and there are lots of traps that are being developed to try and replicate this, and some work, some don't work as effectively. But this is basically just a capture of sort of the environments that we are working with, and we will be working to support. And that's basically all I wanted to chat about. Hopefully I didn't go too fast, and I was hoping to get some discussion up and running from that.
That was great, Des. Absolutely perfect to sort of frame it and then bring it back to ways in which Tupaia can support. Yeah, I guess now we've got about 10 minutes left to throw it open to the floor, and yeah, I guess see if there's any questions, either technical or looking for good stories. We're just looking at that. He's sucking them up with his mouth, right, Jess? Through that. That's amazing.
Yeah, he literally will wait for them to land on his leg, and then he sucks them up in that straw, and there's a little cup here next to him, and he'll spit them out into the cup, and they'll change that probably every hour.
It's amazing.
Yeah.
I love it.
And they actually.
How far can a mosquito fly?
Oh, very good question. So it depends on the species. So Anopheles mosquitoes can fly a little bit further than Aedes. So but again, Anopheles is the genus of species. So there's lots of different species within Anopheles. Roughly 3 kilometers, they say, for Anopheles, but Aedes mosquitoes can be within a few hundred meters generally.
Their entire life will be spent within a few hundred meters.
Yeah, basically. And so, and that's actually interesting because Aedes mosquitoes, so dengue is largely considered sort of an urban disease, and it's because they breed in small, really small places like coconut shells or tires, and they like to live near, or they need to live near humans more than Anopheles, or closer to humans. They both generally need humans to eat and feed, but they live closer to us.
And is it true that the female needs a blood meal before laying eggs? That's why they challenge.
Yes. Yeah, exactly. And so basically it's the female mosquitoes that will bite us and transmit the relevant pathogens. Yeah.
And what's happened to that they were breeding mosquitoes that don't have any or something? Is that?
Yeah, that's still going. So I think you're talking about the Wolbachia project, which is really cool. So the World Mosquito Program is a organization that breeding basically genetically modified mosquitoes. This is made mainly for Aedes. And so what the Wolbachia project are looking at are breeding effectively or introducing a bacteria that's transmittable between Aedes mosquitoes that makes those mosquitoes sterile. And so that effectively to try and wipe out the Aedes populations in priority areas. So it's being rolled out. The challenge is there's lots of, it's not the first time these type of things have been done, but this, I think, whatever biological reason, this one has people say this has a bit more of a promise, but I think from a resource perspective, it's hard to maintain these populations. You've got to grow them in a lab, you've got to feed them, you've got to keep them, you've got to monitor them. And again, the mosquitoes are super tricky. They have a habit of sort of working around those type of biological controls. They're very good and they have their place, but it's not the only solution.
I've heard that said before, Jez, that there's no silver bullet. You've kind of got to be doing everything. You've got to be doing Wolbachia, you've got to be doing insecticides, you've got to be doing bed nets all at the same time.
Yes, absolutely. And there's no shortcut in that sense too. So, which is super fascinating. Basically, the trick is we do have tools to manage and to do really good vector control. The key is to doing it well and being able to account for whether we do it well or not is really difficult at scale. And so in the 1950s, what I found super interesting when I was researching this stuff, in the 1950s in Africa, WHO literally almost eradicated malaria from Africa through really high, intense, focused interventions, and that was doing really good geographical reconnaissance, a really good GR to get these really good baselines. And when they sprayed, they didn't miss a house. When they gave mosquito nets, they didn't miss a household. Whereas when I first entered the Pacific, we were getting sort of spray coverages of villages that were basically around the 50% mark, less than 50%. So often, people just did not know, and we know effectively to do an effective spray campaign, for example, you needed way over 85% spray coverage, and so often the trick is if you're going to do something, you do it well, and we need good tools and good data to do that, obviously.
Jerry, are there any other adverse outcomes of spraying the wrong insecticide? Like, does it kill any agriculture?
Yes and no. There are. So often, well, again, one of the most effective insecticides that have been used in the past was DDT. It's not used anymore, obviously, because it's got some great environmental impacts. And so, yeah, it is something that is considered, but the insecticides that they're developing for vector surveillance, I think, are engineered in a way that are designed to minimize those impacts. I was going to say something I forgot. Oh, yeah.
On human health, Jez.
Well, again, I don't 100% know. They're effectively safe, absolutely. But again, I think there were issues with the bioaccumulation impacts, particularly with DDT in the past, but that's just not, it's not so much an issue anymore. And so basically bioaccumulation is little insects take this, consume this insecticide, the bigger insects, bigger fish, human, and eventually lead to humans, and the insecticide doesn't break down, but that's no longer the case with the insecticides that you're using. But don't quote me on all of these things, because I'm not an expert on the types of insecticide classes, but they're effectively safe, as opposed to what was done in the earlier on in the 50s and stuff.
The am I muted? Yeah, with the mosquitoes becoming resistant to certain insecticides, is there any sort of accurate surveillance at the moment around how quickly that happens, like over how many years and or anything like that? And is that something we're going to capture into Tupaia at all?
I don't know. That's actually a really good question. I don't know sort of the time frames around this. I think a lot of the places we're still trying to work out what insecticides they're actually resistant to already. So it would be something I'd have to look into sort of the time frames. I get the vibe. It's not super long. Like it's not like overnight, but it's not 15 years type thing. Or could, yeah, I'm not 100% sure, but that's absolutely something that could be looked at over time. And so when we're collecting this sort of temporal data, we should be able to see that, or see the sort of both sort of the temporal change over time, but then the spatial distribution, so we can actually see the spread of these insecticide the spread of insecticide resistance as well, which is fascinating and real big issue in Southeast Asia in particular, because a lot of these multi-drug resistant parasites and vectors spurn for some reason, and they don't necessarily know, out of the greater Mekong region, and kind of spread outwards from there. And so that stuff would be super cool to look at.
Gary, that predictive model you had was quite cool, of showing the new potential areas of that could develop local transmission. Is that something that there's any plans to do with Pacmozy or Strive within Tupaia?
Yeah, it was some of my favorite stuff that we ever did. I really enjoyed it. Probably, so there's different ways you can do this. So you can measure vectoral capacity, which is something that has been discussed. And so and from there you can measure things called transmission receptivity. So how likely is an area, or what's the potential for transmission within an area as opposed to another one? Not necessarily things that have been discussed at this stage, but it's certainly stuff that we're collecting the right type of information for, particularly when we're talking with Strive, because we're collecting case-based information as well, that we could potentially be looking at later. I think there's no reason why we shouldn't be.
Oh, my follow-up to that was, were those predefined areas that you then calculated a risk factor for, or was it actually finding the geographic sort of foci itself and putting a bubble around?
Okay, so yeah, basically what was generated in that GIS was based on the spatial and temporal distributions of positively reported cases, and adding a radius that was assumed to be the flight range of a mosquito. So it's relatively crude, but basically, so they changed, those areas changed based on where the cases were being found. And basically, you basically created, so a positive case was found here, 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 and here. And you basically, again, it was a 3-kilometer radius, because we were assuming the maximum flight range of the mosquito vector that was there. And that would create a buffer from that area to basically sort of kind of assume that transmission foci based on flight ranges of mosquitoes and positively reported cases.
Well, that takes us to 12:45, so I'm going to jump in before anyone else asks any questions. But of course, if there are any more questions, Jez is always around to humor. If you spend long enough working in the Pacific, eventually you get malaria. I've had malaria twice, PF malaria, and it's not a lot of fun. So the efforts, over the years to reduce the incidence of malaria, I think are amazing. It's a really horrible disease, but if you couldn't access treatment, which is a whole other conversation, it would be an unimaginable experience. Thank you, Jez. We'll post this up online, and hopefully this is just the first in a long series of similar presentations.
Cool. Thank you, everybody.
Thank you.
Thanks, guys. That was great.